Good morning, class. Today, we will be diving into the intricate field of neurology, specifically focusing on the debilitating neurodegenerative disease known as amyotrophic lateral sclerosis, or ALS. ALS is a complex disorder that primarily affects motor neurons in the brain and spinal cord, resulting in progressive muscle weakness and ultimately paralysis. This disease has garnered significant attention due to the prevalence of its devastating symptoms and the urgent need for effective treatments. Now, let's begin our exploration of ALS by delving into the topic of excitotoxicity. Excitotoxicity refers to the process by which overactivation of excitatory neurotransmitters, such as glutamate, leads to neuronal damage and cell death. In ALS, there is evidence of enhanced glutamate release and impaired glutamate clearance, resulting in an excessive buildup of glutamate in the synapses of neurons. This excessive glutamate accumulation leads to the activation of glutamate receptors, specifically the N-methyl-D-aspartate, NMDA, receptor, which triggers calcium influx into neurons. Calcium dysregulation further contributes to oxidative stress, mitochondrial dysfunction, and neuronal death. Excitotoxicity plays a pivotal role in the selective vulnerability and degeneration of motor neurons in ALS. Moving on to our next topic, let's discuss proteostasis in ALS. Proteostasis refers to the maintenance of protein homeostasis within cells, ensuring proper protein folding, assembly, and degradation. Disruptions in proteostasis have been implicated in various neurodegenerative diseases, including ALS. In ALS, abnormal protein aggregation is a hallmark feature, with misfolded proteins such as TAR DNA binding protein 43, TDP43, and superoxide dismutase 1, SOD1, forming intracellular inclusions. These aggregates interfere with cellular functions and disrupt normal protein degradation pathways. Furthermore, impaired clearance of misfolded proteins through the ubiquitin proteasome system and autophagy further contributes to protein accumulation and neuronal dysfunction. Understanding the mechanisms underlying proteostasis disturbances is crucial in developing targeted therapeutic strategies for ALS. Let's now explore the intricate molecular pathways and signaling cascades involved in ALS pathogenesis. ALS is a multifactorial disease involving several molecular pathways and signaling cascades. Excitotoxicity, protein aggregation, inflammation, oxidative stress, and mitochondrial dysfunction are prominent pathological processes in ALS pathogenesis. The dysregulation of these processes leads to a cascade of events, including impaired axonal transport, disruption of cellular calcium homeostasis, activation of neuroinflammatory responses, and generation of reactive oxygen species. The interplay between these pathways contributes to the progressive degeneration and loss of motor neurons observed in ALS. Now, let's shift our focus to the role of astrocytes in ALS. Astrocytes, a type of glial cell in the central nervous system, have emerged as crucial participants in ALS pathogenesis. They provide essential support and maintain the environment necessary for neuronal survival and function. In ALS, astrocytes undergo significant changes that contribute both to disease progression and neuroprotection. Diseased astrocytes exhibit altered gene expression profiles, release toxic molecules, and fail to support the health and integrity of neighboring motor neurons. However, studies also indicate that certain subpopulations of astrocytes possess neuroprotective properties and may play a role in neuronal recovery. Understanding the heterogeneous nature of astrocyte populations and their distinct functions in ALS is vital for developing targeted therapeutics. Let's proceed to our next topic, which explores the relationship between genetic mutations and the phenotypic variability observed in ALS. Genetic factors play a significant role in the development of ALS, with various mutations identified in genes such as SOD1, C9orf72, TARD, FUS, and others. These mutations are thought to disrupt normal cellular functions and lead to motor neuron degeneration. Importantly, different genetic mutations give rise to distinct ALS phenotypes, highlighting the complexity of the disease. Phenotypic variations can manifest in the age of onset, disease progression, involvement of additional neurodegenerative features, and survival duration. Studying the relationship between specific genetic mutations, their impact on cellular pathways, and the resulting phenotypic heterogeneity is crucial for personalized medicine approaches and developing targeted therapies. Now, let's explore the abnormalities in RNA processing that contribute to ALS pathogenesis. RNA processing abnormalities have been recognized as crucial contributors to ALS pathogenesis. Dysregulation of RNA metabolism, including transcription, splicing, transport, and translation, leads to the accumulation of misprocessed RNAs and aberrant protein production. Specific RNA-binding proteins, such as TDP43 and FUS, are implicated in ALS due to their involvement in RNA processing and their aggregation in ALS-affected neurons. Dysfunction of these RNA-binding proteins disrupts gene expression profiles and impairs crucial cellular processes. Understanding the mechanisms underlying RNA processing abnormalities in ALS will provide valuable insights into disease mechanisms and potential therapeutic targets. Let's now delve into the complex process of neurodegeneration and motor neuron loss in ALS. Neurodegeneration and motor neuron loss are prominent features of ALS pathology. Gradual degeneration of upper and lower motor neurons disrupts the communication between the motor cortex, brainstem, and spinal cord, leading to progressive muscle weakness and paralysis. Multiple cellular and molecular mechanisms contribute to motor neuron loss, including excitotoxicity, protein aggregation, impaired axonal transport, mitochondrial dysfunction, and neuroinflammation. Dissecting these mechanisms will enhance our understanding of disease progression and aid in the development of neuroprotective strategies. Moving on, let's discuss the various disease models used in ALS research. Animal models and cell-based models are pivotal tools in studying ALS pathogenesis and testing potential therapeutics. Transgenic animal models overexpressing ALS-associated mutant proteins, such as SOD1 or TDP43, mimic specific aspects of the human disease. Cell-based models, such as induced pluripotent stem cell, IPSC, derived motor neurons or microfluidic systems, allow for the study of human-specific cellular and molecular events. These models enable researchers to investigate disease mechanisms, test drug candidates, and explore personalized medicine approaches. Now, let's discuss the importance of biomarkers in ALS research. Biomarkers are essential tools in diagnosing, monitoring, and predicting disease progression in ALS. Currently, ALS diagnosis relies on clinical symptoms and electromyography, which may delay diagnosis and limit early intervention. Identifying reliable biomarkers, such as genetic markers, imaging markers, and biochemical markers in cerebrospinal fluid or blood, holds great promise for improving diagnostic accuracy and enhancing patient care. Biomarkers also play a crucial role in monitoring disease progression, evaluating treatment efficacy, and facilitating clinical trial design. Now let's shift our focus to advanced pharmacotherapy for ALS. Treatment options for ALS have historically been limited, with only one FDA-approved medication, riluzole, available. However, recent advances in understanding disease mechanisms have fueled the development of novel therapeutic strategies targeting various molecular pathways. Pharmacotherapeutic approaches include targeting excitotoxicity, mitigating protein aggregation, modulating inflammation, enhancing mitochondrial function, and restoring proteostasis. Gene-based therapies, such as antisense oligonucleotides and gene replacement strategies, also hold promise in addressing specific genetic mutations. Now, let's explore the influence of environmental factors and gene-environment interactions in ALS susceptibility. While genetic factors play a significant role in ALS, environmental factors and gene-environment interactions also contribute to disease susceptibility. Prolonged exposure to environmental toxins, occupational hazards, lifestyle factors, and geographical variations have been implicated in ALS development. Furthermore, interactions between genetic variants and environmental factors can modify disease risk and clinical presentation. Elucidating these interactions will aid in understanding disease heterogeneity and may lead to preventive strategies targeting modifiable environmental factors. Lastly, let's discuss the future directions in ALS research. Emerging areas of ALS research include exploring non-cell autonomous mechanisms, investigating epigenetic modifications, unraveling the role of the immune system, and developing innovative therapeutic strategies. Advancements in gene editing technologies, stem cell research, and gene therapy hold great promise for the development of precise and personalized treatments for ALS. Interdisciplinary collaborations among scientists, clinicians, and industry partners play a vital role in translating basic research findings into effective clinical interventions. Now, let's summarize the key points we've discussed today. In conclusion, ALS is a devastating neurodegenerative disease characterized by progressive motor neuron degeneration. Excitotoxicity, proteostasis disturbances, molecular pathways and signaling cascades, astrocyte dysfunction, genetic mutations, RNA processing abnormalities, neurodegeneration, disease models, biomarkers, pharmacotherapy, gene-environment interactions, and future directions are all critical aspects of ALS research. Understanding these intricacies is vital in developing effective therapies, improving diagnostic techniques, and ultimately finding a cure for this debilitating disorder. Thank you for your attention, and I encourage you to explore further into these topics to deepen your understanding of ALS.